Morning, everyone. A huge pleasure to be here. I'd like to start with a story, actually, a rather sad story, in that, where are we? About 3 or 4 weeks ago, my brother phoned me saying that he wasn't getting any answer from phoning my mum, who's 96. And further investigation and a neighbour looking through the letterbox revealed that she'd had a fall, and he went up there, and to cut a long story short, she was admitted to hospital, completely confused, thinking my brother was her boyfriend, wanting to go home to her mum, and in the final stages of quite, as we now realise, quite advanced dementia. So whereas in the past, when I'd given talks such as this, and I'd said, yes, it's really bad for the carers because they're going through a sense of bereavement, a sense of loss, someone they were really close to who is still breathing but no longer there. At a visceral level, I now know what that is, and I know how devastating it is, and I congratulate you for choosing this career path so that perhaps not in my lifetime, but perhaps your grandchildren will say to you, well, was Alzheimer's ever a problem? You know, if it was a bit like smallpox is now, or polio, well, of course, they're bad conditions, but they're contained and they're no longer the spectre that they used to be. So bear that in mind when you have grandchildren, that they're going to ask if Alzheimer's was ever a problem. So this is the challenging outlook. And we know, as you've just heard very eloquently from Fiona, it's a growing problem. And I'm sure you're familiar with these statistics. We don't have to really dwell on them. Now, why is there so long to develop a treatment? You'd have thought, with it being such a pressing medical need, with the gazillions that's being put into it in the pharmaceutical industry, how come there hasn't been an answer by now? And well, these are the 3 reasons, I think. Forgive me for saying, but although amyloid is indeed a very important component of the neurodegenerative process, I think there's been too much focus on it to the detriment of other approaches. I think the pre-symptomatic time window of 10 to 20 years even, where I could have it now, heaven forfend, where you're not aware of any problem, but slowly, the brain cells are degenerating. And third is a more scientific point, but I gather most of you are scientists. Those of you who aren't, forgive me, that there's the assumption all brain cells are the same, all brain cells are generic, one size fits all, and any neuroscientist will tell you that is not the case. And one of the big questions is, are certain brain cells specifically vulnerable to Alzheimer's? And if they are, what's so special about them? And we'll come to that in due course. So let's look at these 3 issues then. Too much focus on anti-amyloid drugs. We all know what amyloid is, and we all know about the anti-amyloid drugs that are coming. I've always been a voice of not dissent, but of just putting my hand up, just to kind of restrain enthusiasm sometimes. And it was 2 years ago I did this interview that you can still see a transcript of the interview where I said there was too much of an obsession with amyloid. And I want to stress that's not to say it's not important, it's not a significant component. It's just that if you look at the literature, first of all, if you reduce the burden of plaques with drugs such as this one, you're not necessarily going to have any change in cognitive prowess. But more importantly, is in the early stages of Alzheimer's. You can see here stages 1 and 2. This is from Uematsu. There is 0 amyloid in the early stages of Alzheimer's, or there is a pathology. So how can it be the primary target if things are going wrong and there is no amyloid? So I think it's a downstream marker, a downstream issue that can be tackled, and tackled with some effectiveness therapeutically, but I don't think that should be the thing that we are necessarily putting our hopes on, that targeting it will give you a cure or an effective treatment. We've looked already at this time window. This is pre-mild cognitive impairment, before you are deficient on the NMSE score. And I think time and again people are talking about the important need to have some kind of readout that isn't behavioral, that is a biomarker of some sort that will tell you the problem is already underway. This should be differentiated from genetic risk factors. This should be a readout of something that tells you the cell's already dying. And it should be in some kind of body fluid that can be accessed relatively painlessly and cheaply and easily. This is from, I'm sure you work a lot with Alzheimer's Research UK. There's a lot of promotion now of the need to get early diagnosis. I won't read through this. By the way, if you want this talk, I'm sure the organisers will let you have it, and you can look at it in more detail. And only on the news today, I'm sure you all heard, artificial intelligence has delivered a possible diagnostic test for Parkinson's disease, where they've just looked at different profiles of constellations of proteins in the blood and found that you could differentiate pre-Parkinson's from controls, and AI, with the wizardry it has identified this. There's no rationale for why those proteins should be elevated or whatever, but it is a start, and it is at least getting into the public consciousness, I think, the need for a pre-symptomatic diagnosis if ever we're going to stand a chance. Otherwise, you are indeed, for both those degenerative conditions, closing the door after the horse has bolted. More recently was the ApoE test, which was held with great excitement. The only problem is not everyone has the genetic mutation that would give you. And moreover, it's not a direct cause. As far as I understand the ApoE problem, it's that you are at a higher risk of cardiovascular attack, which indeed is a predisposing factor. Again, it's not to say that that's not a problem. It's not being solved by this kind of test. But on the other hand, I think if we want to have a much more wider catchment, we should carry on looking, and that's what we do. Third is this issue of cell selectivity and the assumption that one size fits all, that all neurons are generic. And so any mechanism, to my mind, that is at the level of ApoE or inflammation is treating all cells as though they're the same and ignoring the fact that only certain cells are vulnerable, primarily in Alzheimer's. So one size doesn't fit all because they have different properties. And we know neurodegeneration is selective at these early stages. And surely, if we understood more about the primarily vulnerable cells and what differentiates them and why they're special, then we could have a handle on really understanding how Alzheimer's occurs in the first place. And when we do that, if we do that, then we can get a better treatment for it, surely. Okay, so let me introduce you to the primarily vulnerable cells, which have been around in the literature since 1981 in a really prescient review by someone called Martin Rosser, and that was echoed in the Lancet. And you're all scientists, so I'm sure you're familiar with these kind of pictures. The primarily vulnerable cells, we call them the hub cells, but there are other names for them. Someone else calls them global cells because they act like fountains in the brain with very diffuse outward projections. He calls them, Rosser, and this is a phrase that's now increasingly being adopted, slightly jaw-breaking, isodendritic, meaning that the dendritic arbors of the cells are tangled and wide. We know they release dopamine and transmitters themselves. They are not laminated. These are very basic cells that underlie, as you'll be aware, sleep-wake cycles and arousal levels and mood disorders. They're a very primitive part of the brain. And these, there's very persuasive evidence, are the first to degenerate in Alzheimer's. And this is a really good review I'd recommend, not our work at all, but a very comprehensive overview, making a very strong case that these isodendritic cells are the first ones to degenerate. So if that's the case, what makes them so special? If we knew that, then perhaps we could start to have more insight into the mechanisms of degeneration. Well, let me introduce you to a heresy, and this has been my life's work, although I can't claim it was me that pioneered the idea in the first place. It was when I was a graduate student that I was introduced to this notion. I wasn't a graduate student in 1969, but almost. Oh, no, you laugh, almost. It wasn't that. It was within 10 years I started. This idea, this really crazy idea, that acetylcholinesterase, which you'll all appreciate, is a membrane-bound enzyme that hydrolyzes the transmitter acetylcholine. This is the first thing you ever learn when you're doing biology at the neuromuscular junction. So that's cholinesterase. So why the evidence that it could do something else? That seems really weird. Well, this idea was first mooted by Karksmar and then amplified by Anne Silver in this book, and I think it's really important we always look at the past, and otherwise you reinvent it all the time and waste time. And what Silver showed, as did Karksmar, was that in many places in both neural and non-neural tissue, you have large amounts of acetylcholinesterase, but no cholinestyltransferase, and hence no acetylcholine. So why should nature want to make a huge glycoprotein and not have its substrate there? That's just wouldn't make sense. So could it be doing something else? And I came and joined the fray. You can see here, I did my little review in Trends in Neuroscience back in '84, based not only on the disparity, but also on something else that we found interesting. This, by the way, has been taken up, and I think now people accept, although they can't quite explain, that acetylcholinesterase could have a function that wasn't enzymatic. That's not to say it's not an enzyme, of course it is, but it can do something even when there's no acetylcholine around. So what could that be? Well, if we go back now and look at our critical cells, here they are, you can see that in terms of transmitters, they're heterogeneous. They're all chemically cousins to each other, they're all amines and so on, and you'll recognize them as the very basic, very well-established transmitters. But there's something very interesting about them, and there's something they have in common, And this was shown, for example, the locus coeruleus, which was one of the primary sites for Alzheimer's degeneration. Again, back in the past, Albanese and Butcher, look here, acetylcholinesterase is one-to-one corresponding pretty much with noradrenaline. Why should it be co-localized with noradrenaline? Similarly, in the Nigra, of course, related to Parkinson's, this is our own work, overlapping with tyrosine hydroxylase, the synthesizing enzyme for dopamine. Why should it be doing that? There's very little cholesterol-transferase in the Nigra. Well, the answer, and what I did for my graduate work and subsequent decade or so, this is decades of my work summarized in about 3 slides, was to show that against all the odds, acetylcholinesterase could be released. I will say that again. Acetylcholinesterase, a membrane-bound enzyme, can be potassium-evoked, calcium-dependent release. Therefore, it can act as a signaling molecule. And that was first mooted here when I worked with Rodolfo Linas in New York. And you can see here, these are guinea pig brain slices, living brain slices. You can keep slices alive, as I'm sure you're aware, for about 12 hours. And what we did was to identify the substantia nigra, in this case, with retrograde transport of horseradish peroxidase into the ipsilateral striatum, so you can see it there marked. And then overlapping with it, like cat's eyes here, glowing in the dark, this was a light-emitting reaction that we'd developed for online release of cholinesterase. And you can see here the cholinesterase is released. So could this be a signaling molecule? And if so, could it be important in Alzheimer's, given that it is common to all the cell groups, irrespective of the transmitter, in which we find this phenomenon occurring? Well, obviously, a huge molecule is very unlikely that it's going to actually be a signaling molecule in its entirety, preserving all its tertiary structure. And indeed, and again, in the interest of time, we'll cut the story short. This is the answer to the salient part of the molecule that underlies its non-cholinergic behavior. And it's a 14-mepeptide, which has an eerie correspondence, as you can see, with amyloid precursor protein, and we call it T14. Not a very imaginative name, because it's 14. I mean, now this is going to feature all the time subsequently as we continue together looking at a possible novel mechanism that's driving Alzheimer's disease. So let's think about T14 again in these primarily vulnerable cells. Is there anything else special about them? Just to recap, they are primarily vulnerable in Alzheimer's. They all contain acetylcholinesterase independent of its catalytic function, and they all have this 14-mepeptide that is cleaved from acetylcholinesterase present. Okay, so what could be the final thing that we then need to feed in? What's really interesting, and as I'm sure as early career people, you have the same, you suddenly have a light bulb moment simply by reading the literature. And I can't stress enough how just by looking at what other people have done and then joining up the dots is vital, and it's also really exciting when you suddenly see the light bulb. And for me, the light bulb was this, when I read this review from Nancy Wolfe, and she showed that at 4 weeks gestation, the brain is divided into 2 plates, the alar and the basal plate, and lo and behold, the cells from each of those already at 4 weeks show very different properties, and you probably can't read this at the back. I say you're very welcome to the talk if you wish. And what excited me when I looked at the so-called global cells, she was calling them, because they're like fountains, they have very diffuse projections, as I said, they're very basic, was the fact that they had retained a plasticity, unlike their alar plate counterparts. They retained the ability for development, for growth. And what excited me was, of course, they're all the cell groups that are lost in Alzheimer's. Now let's put those things together. These are the primarily vulnerable cells. They come from a different part of the embryo. So at an early stage, they might have very different properties. And could those very different properties have something to do with the fact that they are prone to degeneration? And could one of those properties be the fact they all have a common chemical, a peptide derived from cholinesterase? Obviously, with rhetorical answers, it is yes. And it turns out that T14 is indeed promotes growth. So here you can see it in the developing rat brain. Those of you familiar with Westerns will recognize them here. So at embryo 18, we have a high amount of T14. This is it quantified in the postnatal. That actually reflects the large-scale neuronal activity in this area of the brain. These are the isodendritic core cells, as we monitor it with optical imaging using voltage-sensitive dyes. So you can see that early on in development, the T14 is more active, and that correlates with enhanced activity because the T14 is enhancing calcium entry, which depolarizes the cells, so it kind of all fits. If you then look in the human adult, in the brain, this is from our own group, you can see here, these are healthy, well, they're not healthy anymore, they're dead, but they didn't have Alzheimer's, they're controls. You can see that even between 37 and 83, we're a decline in this agent that would normally be promoting growth. Interesting still is if you look at other cells that have the same embryological provenance as these cell groups, and that is the skin, the epidermis of the skin. I'm sure you're aware, and it's increasingly fascinating, there are certain links now being made with neurodegenerative disorders and the skin. Bear in mind, the skin is in constant renewal, and we were therefore very interested in seeing whether this T14, which characterized and was in the brain and seemed to diminish with age, if a similar pattern could be seen in the epidermis of the skin. We did this with Unilever, who, as you might imagine, are very interested in skin and aging. And you can see here, we published this in the journal Aging last year, 23-year-old, 30s, and then 50s. You can see that there is a decline. Hope you can see the T14 immunoreactive keratinocytes in the epidermis, which are declining with age. So this seemed to fit. And then in a pilot study, we looked at sebum and saliva. The pilot was in the lab, and I should just say, you might wonder why there's no error bars on the oldest group. And that is because that's an N of 1, and that's me. Because Neurobio is a very young company, and so they very kindly said 46 plus, and let me reassure you that was being generous by several decades. But nonetheless, you can see, even though it's a pilot, we haven't published it, that there is, T14 does seem to be in a lot of tissue, and in all cases, both rodent and human, is reflecting aging. And so it's declining, as you'd expect with something that normally promotes cell growth. Okay, so far so good, but we're interested, are we not, in neurodegeneration? Now, this molecular jackal can turn to a hide. That's to say, something that promotes growth can become toxic because the amount of calcium going in can actually, if it's excessive, can be taken up into the mitochondria where it compromises oxidative phosphorylation, so you get leakage of electrons, formation of free radicals, and death of the cell. And that's what happens here when we've given high amounts of T14. You can see compared to the very nice neurites being shown by the red arrow that with the high levels, the cells are in pretty poor shape. Similarly, you can change the amount you apply it for. So if you just give it for an hour, we see nice growth of neurites. If we keep it on for 24 hours, jackal turns to hide. But what's really interesting about this trophic toxic axis is not that you can vary it with amount or with duration. It varies with age. And this is not our work. This is Emil and Schramm, you can see here. So this means that what is beneficial and will promote growth in a developing brain, in the context of a mature brain, that same amount, that same chemical, will now turn toxic because it's not mobilized for the neurite in the same way as it is in the developing. It's taken up into the mitochondria. So this, if we put this together, we'll start to have a narrative of perhaps what makes those primarily vulnerable cells vulnerable because they've retained that particular mechanism for inappropriate developmental mechanisms being re-triggered. So is T14 indeed really toxic? Could it be the primary agent? Well, if you look in vitro and in tissue culture, you can see it does indeed promote amyloid and tau. It's upstream of amyloid and tau. Similarly, if you look at ex vivo brain slices where you cut down the midline and incubate one side with the treatment and one not, so it's the perfect control, you can see that that too has elevated amyloid and tau. But I think even more persuasive is work from Gregory Cole, who we collaborate with at UCLA, where he's looked at the 5 times familial Alzheimer model. I'm sure you're familiar with this. This is a kind of gold standard animal model that people use that overexpresses amyloid. You can see in the transgene negative, there is no detectable signal, or very little. But if you look in the actual animal model, you can see T14 immunoreactive plaques characterizing this animal model, differentiated from the control, suggesting it is indeed a toxic agent, even in an animal model. So given that, we've tried to develop our own animal model with the University of Nottingham and a company called Evotech. And so far, we haven't looked at the behavior. We're now getting a license. I'm going to collaborate with Oxford University so that we can actually do in vivo work. But even so, if you give T14 or the wider agent T30, which is its wider molecule, but it contains T14, if you do it twice a week for 3 weeks, then you can see neuron numbers gone down, and indeed amyloid beta and T14 goes up in the brain. So it does seem to be an upstream toxic agent. And therefore, I'd like to suggest to you that neurodegeneration is an aberrant form of development driven by T14. Let's think about that. This is the rat brain, which you've seen. And we know that in the midbrain, the T14 can be activated, it can be released. The blue are DAPI stains, it's nuclear stains, and the green is immunoreactive T14. So you can see it's being released as a signaling molecule, which differentiates hugely from amyloid. Amyloid is not a signaling molecule, it's a sort of thug, whereas T14 is actually an evoked agent that is dependent on neuronal activity. Here again, you can see it correlates very nicely with dopamine and the substantia nigra, which of course is lost in Parkinson's. And in the midbrain here, you can see this is in Alzheimer's. You can see that in the age-matched controls, it's half what it is in Alzheimer's. So this is the midbrain. So the T14 is recapitulating the developmental profile that we see in the rodent. So suggesting that it is indeed the hub of it. Now, if you also look at Braak stage 2 in the Niagara, you can see partially filled T14 immunoreactive cells in the pars compacta region. As you progress to Braak stage 5, you have fewer cells more intensively filled. So if you look at that as a metric, then you're having an overall increase. So this shows you how, I don't know how you feel about the overlap, the co-pathology of Alzheimer's and Parkinson's, but my own view is that T14 is a mechanism common to both, and indeed, as you'll know, the nuclei are adjacent to each other. They're all part of the isodendritic core cells. So could T14 be the active driver of this? Well, what we're suggesting, and this is our narrative, is that in the early stages of life, at Braak stage 0, T14 is there, but low levels, but you still have the capacity, these cells, these cell groups, have the capacity for growth if activated, and there is no amyloid. Now what happens is if there is damage to these cell groups rather than others, and this can be anything, a blow to the head, ischemia, or a decline in protective mechanisms such that it's easier to form free radicals, and we know that is a feature of the aging brain. It's not an automatic consequence, but it could explain why this happens in older people more. Then what happens is these cells can retaliate in a way the others, the alar plate-derived cells, cannot. That is to say, in response to damage, they will try and grow again. And they will do so by mobilizing the T14. Big problem, we've seen the T14 is toxic in the older brain. So now the very worst that it could be doing is trying to compensate in a feedback by instigating a feed-forward mechanism. So now this T14 will cause more cells to die, and that will release more T14, and more will die, and more will die, and that accounts for why we have what we call neurodegeneration in cell groups that have a high degree of redundancy. We know that in Parkinson's, 80% or something of the cells have to go before you get motor symptoms. So because they are so basic, because they are so intertwined with their dendrites, they can take over and they can still function even though they are progressively damaged. And because of that, you have this 10 to 20 year time window. To the best of my knowledge, I haven't seen any other accounts for why there is this time window that we know occurs, and I think it's due to the redundancy of the isodendritic core cells, which in turn are the selectively vulnerable cells, in turn being at the heart of the problem. So this then carries on, and the person is still oblivious. They might start with MCI, and only when it spreads to the hippocampus and cortex, and you have cognitive impairment and amyloid present, only then people present at the clinic. And that's why I think we really do need to look at T14 and try and look at it as a pre-symptomatic marker and then a means of interception at the pre-symptomatic stage. Okay, so if it drives cell growth and renewal, so it can also apply to other things, neurodegeneration, cancer, and we've seen it's all over. Cancer is an inappropriate activation of developmental mechanism, and possibly to hyperproliferative skin disorders such as psoriasis or eczema. But we'll focus on neurodegeneration. And let's look first at the possibility of a readout, just checking time, yeah. You may be familiar with this quote from Bill Gates, and we've already looked at the need for a readout. We disagree with him. We think we want to have a diagnosis not before the symptoms get too bad, but before they even appear, because we've got this time window. So what progress are we making to that? Well, if we look at Braak stages, the asymptomatic stages 1 and 2 in the post-mortem brain, you can see the T14 is now reflecting the degeneration in the post-mortem brain. But really what we want to do is to reproduce that profile in living people. Now, CSF is not the ideal matrix, but it's the most intimate with the brain. So if we can't see a profile change there, we'll never see it anywhere. So we've started off with CSF, although we have some other ideas as to other things that we could sample. And in collaboration with the hospital clinic in Barcelona, you can see we are seeing a trend now for the T14 in living CSF to actually reflect what we're seeing in the post-mortem scenario. Okay, so the advantages, though, of working with living things is that, nonetheless, there's still all the variation of diet and medication and so on, individual variation. So it's better, usually, if you can, to have it to an internal marker where the person has their own kind of internal control. And what we found is that if we look at it as a ratio to tau, then we see something very interesting. You can see here that in the control CSF, living CSF, there is a very high correlation between tau and T14, a significant correlation. What is interesting is if you then look at mild cognitive impairment and Alzheimer's, that disappears. It's gone. Now, that might be an interesting way of differentiating it, possibly. It also suggests, in my thinking, is that there's excessive T14 now being produced, so it throws out that one-to-one lockstep variation. But you could say, well, either way, that's all very interesting, but if they've already had a diagnosis, that's not the early stage. You promised us early stage. Okay, so let's look at the control group and see if that gives us any clues. Now, remember I said that these cell groups, early on, you can have mood and sleep disorders because they have there are the mechanisms, underlying mechanisms of arousal and mood and so on. And mood and sleep disorders are often documented as an occasional, not an occasional, as a frequent prelude to neurodegeneration, but not an exclusive one. I want to stress, if you've been sleeping badly recently or are in a bad mood or you're depressed, it won't mean you've automatically got Alzheimer's, but it can be a predisposing or indication, because if these are the first cells to go and these are the ones that underlie sleep-wake cycles, it follows that if they are damaged in certain scenarios, then you could already have a differentiation. So what we did was to, again, look at these cell groups, just to remind you that they are what I call the amine fountains and the targets of antidepressant drugs and so on, and actually see if we could differentiate the control groups so we could have a truly, not so much a test as a screen. So we found that there were 10 people with a history of mood disorders and 12 without. Their tau was the same. So tau, although it's a great marker, and some might argue more early stage than amyloid, you can see that it's absolutely the same. But look what happens if you do T14 to tau. Although the tau is barely significant, T14 on its own, but as a ratio, you can see there is a significant difference, and that shows a ROC curve, which I'm sure you may be familiar with, of 0.82. Which is significant 0.01. So even though these are very small numbers, this shows that we might be onto some way to have an early stage screen, and I stress screen, not diagnosis, for differentiating people that have cell groups that are disrupted primarily in early stage Alzheimer's. But it could be they're disrupted for other reasons, and I want to stress that we're not saying this is a test for Alzheimer's. It's a test for disruption to the isodendritic core cells. Okay, so could it be a screen? Well, no one wants to have lumbar punctures, but happily it seems that nasal secretions, 50% are derived from CSF. So we could start to think about a lateral flow test in some way. T14, very obligingly, is detectable. We've shown it, as you've seen, it's in blood, it's in saliva and sebum, and these are nasal secretions, again from the group not published. So the T14 is there, and it's in nasal secretions, and if indeed the profile in nasal secretions would mirror that seen in cerebral spinal fluid, then we would have the basis for developing a lateral flow test for a very early stage screen, based not on Alzheimer's, but on disruption of the primarily vulnerable cells. I can't stress that enough. So I was talking earlier with Jess about how really, for a diagnosis for Alzheimer's anyway, you really need it in 2 stages, because if you want to get it at the asymptomatic stage, people aren't going to just go to the doctors, nor would the resources enable that to be possible. You need some home test, like we did with COVID, but you can't market it, nor would you be qualified to say that it was an Alzheimer test. But you could possibly have it as a brain test or mood test or something like that. It's something general that was a screen that would tell you to go to the doctor. So a go, no-go test, so not a highly quantitative one. And then if you go to the doctor, what we're also working on is a saliva test that would then be sent off for mass spec detection, and that would give you a highly quantitative readout, as well as all the other complementary medical evaluations that would give you a proper diagnosis. And then, ideally, you'd have our therapeutic. But it seems to me that you do need whatever a 2-stage process for diagnosis, because you can't have a home test that gives you a definitive diagnosis, even if it were possible. And at the same time, you can't have everyone going to the doctor, everyone over 40 going to the doctor. So how can we intercept it? Well, this is very simple pharmacology that typically one tries to block the receptor, and this is what we're developing with our nasal spray device. So we first had to identify the target. We've done that over the years, and it's the alpha-7. This is some early evidence showing if you block NMDA receptors, you can still see calcium potentials. More persuasively, if you transfect oocytes with the alpha-7 receptor, then you see that in the presence of the primary ligand, shown by the small little dash, there is an enhancement of the current, which means that it's acting at an allosteric site, not at a primary site. That's not the same with butyrylcholinesterase peptide or indeed with alpha-4 beta-2 transfections. And most persuasive of all, in the human brain, there's a very high correlation between T14 and the alpha-7. We published this year a paper claiming that it was exclusively at that site and got away with it, as you can see here. So hopefully, if you then do overexpression on mutations, you can change the sensitivity of the T14 to the receptor. And again, that seems to be very strong evidence that this agent is acting exclusively at an allosteric site on this very powerful calcium ionophore in the brain. Even more persuasive, if you look at the striatum, which has cholinergic receptors but not the alpha-7, you can see there's no real effect at all when we look at optical imaging. However, when we look at the nigra in the same brain slice, which does have alpha-7 receptors, we see a clear effect. So that again suggests a site selectivity. This is just, because of time is short, this is just a summary of what I've just said. I won't read it through word for word. Most interesting is it seems also the peptide to increase its own target. This was done with Nadine Kabani in the States, who we gave some. By the way, if anyone wants any of our reagents, you're very welcome to them. You can see here that's increased the lamellipodia surface area, and also we've shown increases, as she does, the number of alpha-7 receptors. In rat brain slices, again, we can show that there's an enhancement, the peptide's enhancing its own target, which would add to the feed-forward effect. So how can we block it? How can we block this? If I've persuaded you that T14 is driving neurodegeneration and that we've identified the target and it's high in early stage, how can we now come in that pre-symptomatic window, ideally, and block it? Well, we've come up with an idea that's a much more straightforward one. Rather than trying to devise something that acts at the alpha-7 receptor, we've just taken the T14 itself and rendered it inert. So this is called neurobiopeptide 14, and the way it works is to bind to the allosteric site, that's T14 in its linear function. If you cyclate it, then it will displace the endogenous toxic linear T14, and thereby neither can work, neither the endogenous toxin because it's been displaced, nor indeed the drug because it's a cyclated variant. And if you do that, then it seems that it could work. We can see it displaces T14 both in ex vivo brain slices. In vivo, when we inject it, it displaces the T14 maximally at 4 hours. And again, Greg Cogg's work with the post-mortem brain, you can see that the binding, the metric is the binding of the T14 to the alpha-7 is gradually reduced with increasingly dose-dependent concentrations of MBP. Again, showing that that's how it's actually working. It seems to work in the 5-FAD animal model. This was done in collaboration with the CRO Evotec a year or so ago. You can see that animals intranasally given MBP14 twice a week for 14 weeks, they put on weight better than the vehicle controls. Novel object recognition, where an animal will explore an object if it's new, if they've not seen it before. So you can use exploration time as a test of memory for differentiating novel from familiar. You can see here that the wild-type animals explore the red, and shown in red in this case, the novel object much more than the blue, as you might expect, whereas the Alzheimer multiple mice, the 5-FAD, are living out their genetic destiny. They can't differentiate familiar from novel, so they're equal. But the genetically modified animals given our drug twice a week are behaving like the wild-type animals. And if you look at amyloid as a downstream marker, you can see it's substantially reduced in the brains of. Now, if this worked in humans, this is what we're now aiming to do, to try it. This was announced some time ago, and we can actually, I don't have time now, go step by step in how it actually works, which is why it made the front of Sci News. I'm sure, Journal, you will read avidly, but you can see that they were acknowledging that we could actually go step by step in the mechanisms of how the drug was working. Les echoes were very flattering. The MHRA have now given us a certificate of innovation, a passport of innovation, which is a formal accreditation that this is a truly novel approach. And you might imagine we're very pleased with that. Comparing it with the anti-amyloid drugs, hopefully, if it works, it's quite straightforward. But the one that excites me most is the fact that we'd have minimal side effects, I hope, because it seems to act only on one receptor. And also, it could be administered at home. You wouldn't have to go to the hospital and have lengthy infusions. So, to conclude, how far is this new advance, this new kind of approach taken us? Well, I think if we look at the early stages, we can think of a two-step diagnostic, a screen, and then a more quantitative readout. And if that shows that you do indeed have the early stages of Alzheimer's, even though you are cognitively normal, you could then be given a drug that will stop the cells. If it works in animals like it works, if it works in humans like it works in animals, it would then stop any more cells dying. It would halt the progress. So you'd stay at the pre-symptomatic stage, and that means you'll never get the symptoms. That's our dream. And it's something that could be rolled out, I hope, in the NHS. It wouldn't need complex brain surgery or ethical worries about stem cells or anything that was painful or expensive. It would be quite straightforward. It would be a routine test you bought in the chemist that could be available on the NHS for poor people, possibly. Trip to the doctor, saliva test, and then the prescription of a nasal medication twice a week or 3 times a week, possibly, if it was like mice. And then you do that for the rest of your life. And if that happened, then indeed your grandchildren could come to you and say, Granny or Granddad, have you taken your nasal spray today? They could say to you. And was Alzheimer's ever a problem? They would say. Thank you very much. By the way, so if anyone wants this talk, just ask the organisers. And if anyone who's researching in areas like this, we really want to give people antibody and T14 and the drug, because the more we can get external validation, the more happy we are about this. So if anyone would like to contact me, do.
And that was just extraordinary, and I think it just started blowing everyone's minds. I mean, there's so much in there, and I suspect we'll talk on the train, but I'm really struck about the barriers to kind of getting this out.
Money for your
yeah, but I was also thinking about kind of the ethics and moral piece around the kind of the screening piece and kind of going out.
Sure.
That bit. What you say money, but what do you think are the biggest barriers?
Okay, so I think a screen that was simply telling you that certain brain cells were on the blink a bit, yeah, with no mention of Alzheimer's, no mention of would be comparable to me wearing a Fitbit telling me my heart rate's unusual or something like that. And I personally, I don't see why that would be ethically a big problem, as long as you won't make any claims, as long as it was just saying, you're just checking if you're in the normal range, if things are working. If they're not, go and see the doctor, a bit like my Fitbit. If my heart rate was wrong, it wouldn't tell you I had, I don't know, aortic block or anything. It'd say, go and see the doctor, yeah? So I think ethically one could pioneer something like that, and as long as one didn't make extravagant claims. And we've had a quote from a lateral flow company, because their order books are empty at the moment, Artica, and within it's cost £50,000 would take 6 months to have the kit, to hold it, to develop it and validate would take longer. And we'd seek advice from people like yourselves on how one validates things clinically and so on. But that is very reasonable. On the saliva test, we're working with the Rosalind Franklin Institute in Harwell, who are, they position themselves as physical sciences in the service of biology. Yeah? And we were awarded an internship where one of our team could go there for 3 months and try and get detection of saliva using mass spectrometry on a machine that is widely available. Yeah? And then it's just a question of, you'll tell me how one would then get approval from the FDA. And you saw, I mentioned GNL, who are our regulatory partners in the States. Hopefully we could prepare an application to the FDA or the MHRA, because no one dies giving a nasal swab. So if that could be validated. The first thing we need to do is to show that what we're seeing in CSF is mirrored in the same person in nasal swabs or in saliva. So what we need is to engage with biobanks that have samples of both. The other thing that's very exciting is any biobank that has historical samples, so samples going back 10 or 20 years, okay? And if they've been carefully documented and sequentially sampled, say, every 3 years, what you need is to look at the control group from 10 or 15 years ago. And if you can differentiate, using your test, blind, that in this control group, some are different from others, yeah, showing a very different pattern, that it was the ones that are different that then went on to convert to Alzheimer's, that would be proof that it could be used as, and that's what also we're very interested in doing. We have from ABLE, which is a blood bank in Melbourne, we have very precious blood, we're waiting to get our detection systems really good, but we want to look at the historical samples of, did the controls, the ones that go on to convert to Alzheimer's, already show a difference 10 years, and that would be, I think, very persuasive. So that's the kind of what we need on the biomarker. On the drug, it's good to go through the IND stages now, and that's just a financial thing. So we've started, we now have what's called API. We've got bulk production of it. So we have 30 grams, if you want, we've got 30 grams of MBP weighting at research grade, not clinical grade. It's performing well in the toxicology studies we've had done so far, but as you'll know, there's a whole battery that has to be done. It has to be in a species, not a rodent, and then we just have to commission that. So once we have funds, we can do that. And so with a fair win, within a year or two, we could be in phase 1, we hope, with that. Well, I say well, the reason I say money is that, you know, we have now, and for those of you who haven't seen it, we had a review published in Alzheimer's and Dementia about 3 weeks ago, And it was a review partially authored by people that weren't in the group but were Alzheimer clinicians. So we feel now people are gaining traction. But our problem has been, because this is so different from what other people have done, people mistakenly think I'm being rude about amyloid, and I'm not. It's just, I see this as, it's been much harder to convince people and to convince the world. Yeah, but it was ever thus, as you'll know if you've done research. Yeah.
Sorry, my hand raising, so I'm so excited about this. Given the sort of non-invasiveness of your measures as well as the potential therapeutic application, would there be some scope and possible maybe kind of fast-tracking potential to apply this in models of Alzheimer's disease like Down syndrome, where people are showing earlier diagnosis, faster progression, and are looking for non-invasive assessments and intervention? Have you considered applying it in Down syndrome or learning disabilities?
Totally, yes, absolutely. And I think that has special fast tracking because it's, young people, very much so.
Thank you very much, Baroness. That was really interesting. I'm a person living with Alzheimer's, third generation. I'm nowhere near a scientist, so I thank you for the great lesson. A very interesting idea about the T14, T30. My question is, why does it turn toxic?
Okay.
And then secondly, a lot of people say now you can prevent Alzheimer's by eating spinach today or eating strawberries tomorrow.
Yeah.
Can the T14, is there anything you can do to prevent it?
Okay. Thank you for that. So I'll take the question first. The T14 is toxic because it works at a target that lets calcium into cells, a very well-known, very powerful so-called receptor that lets calcium into cells. In its proper time in development, that calcium is then mobilized for the cells to grow. However, because of the complexity of the intracellular situation, in a mature cell, that doesn't happen. So you've got all this calcium sloshing around, and it's taken up into the powerhouse of the cell, the mitochondria so-called, because it just all sucks it up. And when that happens, then the cell can't produce energy in the same way as it would normally do, because there's a very fancy thing called a transport chain where you have energy providing the cell. That is compromised. So all these little electrons kind of fall out, and they stick onto oxygen, for example, and form things called free radicals. And they are like molecular Rambos that eat away the cell, so the cell dies. So that's how it's toxic. In terms of eating, this is almost a planted question, I feel, because we've been discussing. The reason I asked if you're Australian is I was recently in Sydney about 3 weeks ago as a guest of Neuroscience Research Australia. And there's a lady there working on mitigating factors for Alzheimer's. And one of these is diet and the kind of food you can have in the diet so you're not eating. Now, of course, you can't say this food will stop you getting Alzheimer's. But it struck me that if we can do anything at all that reduces the risk, and I say reduce the risk, not prevent it. So we had this idea, and it's gaining traction here with you guys, of a recipe book, which I can call Food for Thought. I thought this up in the night, and it would, although there's lots of books about brain health and eating, to the best of my knowledge, there's nothing specifically on Alzheimer's and what you might do. And I thought we could ask different people to do recipes. The scientists could make up recipes if we gave them the list of relevant ingredients. So, and this would be, and then the Alzheimer's Society could endorse it. That's the plan. Yeah. So, and you'd get some money out of it. So this is my sideline. But so thank you for that question. But in terms of what you can do, other things, certainly exercise. We know that increases neurogenesis, increases oxygen to the brain, and so on. So that's really good. Diet, it would seem, but that's not my area. But I can let you have, or the organisers will let you have the name of the person working in Sydney on that. And I think also, the more you come to things like this, the more you talk, the more you engage. My mum, for example, my brother and I noticed she was always better after at her sheltered home, after they'd had a party or some kind of gathering. And Graham could tell the next day she was much more lively and animated, and we saw it was because she'd been involved with people, even on a short-term basis. So those things are not going to make you better, and they're not going to cure. But in any event, don't you want to eat nice food and mix with people and and take it? They're things that are fun to do anyway.
I have two questions. The first question is whether you think there are other peptides expressed by other subpopulations of neurons that have a similar toxic effect to T14. And what do you think about the idea that the regrowth of these neurons is causing this effect? And how does that then impact studies that are looking to regenerate regenerating neurons.
Sure, okay.
I didn't hear very well, but the second question I'll do first, because that was studies that promote things like BDNF. Yeah, I think that actually is not appropriate. My own view is that anything that's promoting calcium entry in a mature brain should be done with caution, especially if it's in those cell groups. So that's my own, the jury will be out, time will tell. Is the other, you were asking about other cell groups with T14? Was that your?
Either T14 or other peptides.
Sure, okay, well let's do T14 because what I found really interesting, we've published as a side thing, it's not our core activity, we have to collaborate on cancer, yeah? And what we've shown there, we published this in 2017 with someone called Chris Pepper, and we showed that there were three groups in myeloma, leukemia, and breast cancer, and in each of those conditions, there was a metastatic cell population and non-metastatic cell line, and the MBP, our drug, lowered the migration of the metastatic cell line in all cases to that of the non-metastatic cell line, which was very exciting. And that was published in 2017 in OncoTarget, a journal I'd never heard of. And we've now collaborated with a group, and we're about to publish this, looking at xenograft animals where tumors are implanted into mice. And these are patient-derived tumors, colorectal tumors. And indeed, the MBP, given systemically, not only, again, doesn't seem to do any harm to the animals, they tolerate it really well, it actually significantly reduces the size of the tumor. And if coupled with standard of care, which is chemotherapy, it enhances it, so it could be used as an adjuvant. So there seems to be a lot of possibilities for cancer. And then in the skin, Unilever are very interested, and I'm very interested possibly in topical application of MBP for psoriasis or eczema, for example. And also in the link between, there's some papers out showing a link between psoriasis and Alzheimer's and various skin conditions and Alzheimer's, and the skin has amyloid and tau in it as well. So because they both come from the ectoderm, it's fascinating to see that there might be some parallels. I don't know if that answers your question, but when you say other peptides, we're busy enough with T14. I think that's keeps us going. That's not to exclude other peptides, of course. Yeah.